Hello guys, welcome back to my channel. This is a quick video of the pathogenesis of cholera or Vibrio cholera. So to start with, we must know what is the mode of transmission, how the disease gets transmitted, right? Or how the bacteria gets transmitted. So the main mode of transmission is contaminated food and water. So once we take contaminated food and water, the bacteria enters the body. Then what does it do? Step 2. It crosses the protective layer of mucus in the intestine. Suppose this is the mucosal lining and this is the epithelium. Okay, this one is the epithelium of the intestine. So the bacteria moves from the mucous membrane. It crosses the mucus layer, protective mucus layer, and enters the epithelium. Okay, let's quickly look at the factors which help in the crossing of protective layer of mucus. There are three main factors. Okay, we all know Vibrio cholera shows starting motility, so it becomes very easy for it to enter our intestine. Second is it secretes mucinase and protease enzymes, which also helps in the transmission. Hemagglutinin protease is another important enzyme. What does it do? It cleaves the mucus and fibronectin and releases the fibrose in the epithelial lining. Okay, so what happens after it enters after the Vibrio enters the epithelial lining? It adheres, means it attaches to the epithelial lining. Okay, it adheres and then colonizes. It multiplies. So it adheres with the help of type 4 fimbria. It adheres to the intestinal epithelium, right? And this is the site where it starts doing its action. So in the epithelium, it releases cholera toxin. Cholera toxin is a very powerful enterotoxin and it is heat liable. What does heat liable means? It simply means that this toxin will get destroyed when the temperature is increased. Okay, let's quickly look at the mechanism of action of cholera toxin. So now let us look at this beautiful picture, figure number 31.2 from Apurva Shastri. The cholera toxin has two fragments, namely fragment A and fragment B. Okay, the fragment B is a pentameric fragment. Okay, it has 5 subunits and it attaches with the glycoside receptor. Can you see this red one is the glycosidal receptors? Okay, fragment B attaches with the glycoside receptor. This fragment A enters into the epithelial cells of small intestine. So once the fragment A enters the epithelium, the fragment A goes and binds with the G protein. There it causes ADP ribosylation of G protein and increased activity of adenylate cyclase. Okay, so when there is increased activity of adenylate cyclase, it causes accumulation of cyclic AMP inside the epithelium. So there it goes. What happens if there is increase in cyclic AMP? Yes, it moves the water passively into the bowel lumen. Okay, so once the water moves into the bowel lumen, to maintain the osmolality, it causes accumulation of isotonic fluid and hence watery diarrhea. Watery diarrhea is the final clinical manifestation that we see in cholera. Alright, this was the pathogenesis of Vibrio cholera. You can also note down the gene for cholera toxin, as we all studied before, that the CTX, the cholera toxin gene 1, is for the classical biotype and the cholera toxin gene 2 is for the Eltor biotype. That's it for the video. If you like, please subscribe for more videos and the next part will be updated soon. Thank you.